Now we're going to take a moment to look at the classification and taxonomy of viruses. The important piece to this kind of section here is that you understand that viruses are not classified in the 3 domains of life. Remember that they are not considered living because they don't fit the criteria for something that's alive. They're not composed of cells is one reason, very simply. But their numbers are so great that they do require some sort of classification in order for us to talk about them and to research them and to take a look at them. So since 1971, we have this International Union of Microbiological Societies Virology Division. They are the ones that are going to have been going through developing, refining, and maintaining this virus taxonomy. To the International Committee on Taxonomy of Viruses, so the ICTV. It should be noted that viruses mutate very, very quickly. And since they mutate so quickly, it's very difficult to classify them into a genus-species type of binomial nomenclature, just like we do with bacteria and other prokaryotic organisms, for example, and eukaryotic organisms. So the ICTV's viral nomenclature system classifies viruses into families and genera, based on their viral genetics, chemistry, morphology, and mechanisms of multiplication. So basically when we talk about viruses, we're going to talk about viral family names. Those end in viridae. So when we're talking about a family, you'll know that it's a family because they end in viridae. And then their genus name is going to end in virus. So we have parvoviridae and then parvovirus. So the names of the viral orders, families, and genera are all italicized. So remember, when we are talking about that binomial nomenclature system, we italicize the name, the genus and species. We do the same here with viruses. So the order, family, and genera are all italicized. So when we refer to a viral species, we use the genus and species epithets. So our Pandoravirus dulcis or Pandoravirus salinus. Generally, though, when we'll be looking through things and studying things, we will be looking at the general family or the genus, not very specific species, except in some very specific cases. So we'll be using the general Pandoravirus, for example, when we're talking about these things. It's also noted here that the Baltimore classification system is an alternative. It kind of supplements this ICTV nomenclature. So it classifies viruses a little bit differently. They're grouped into categories based on chemistry, morphology, or other characteristics that they have in common. So categories include naked or enveloped structure. So that's a very easy way to classify things, whether or not they have an envelope or not. Whether they have single-stranded, which is SS, or double-stranded, DS, DNA, or single-stranded, double-stranded RNA, genomes. So again, remember our viruses are very simple. They have nucleic acid, they have a protein coat, which is the capsid, and then they might have an envelope on the outside of that, aside from some other that are a little bit more complex, like the bacteriophage. But the general way that we classify them then can be either naked or enveloped, so whether or not they have an envelope or not, and then based on the genomic information, so based on the genome that they carry. Is it a single-stranded DNA or a double-stranded DNA? Is it a single-stranded RNA or a double-stranded RNA? Also, is it segmented or non-segmented, the genomes? And also, is it a positive strand RNA or a negative strand RNA? So if it's a negative strand RNA, then you have to, the virus will have to take that negative strand RNA, make a positive strand, and then can go through the process of making proteins. So there are different types of mechanisms based on the type of virus. So for example, the herpes virus is classified as a ds-DNA enveloped virus. So that's one way that we classify that, for example. So this would be double-stranded because it's DS. DNA, so it has DNA rather than RNA, and then it has an envelope because it's enveloped. Our human immunodeficiency virus, or HIV, is a positive ssRNA. So positive meaning it's a positive strand, single strand of RNA, and it has an envelope. So positive strand, single strand RNA. So positive single strand RNA enveloped virus. Our tobacco mosaic virus is also that, but it doesn't have an envelope. So We have our positive single-strand RNA virus. So then there are other characteristics like specificity, tissue, capsid shape, and special genes or enzymes that can be used to describe similar viruses, but generally we're going to stick to this positive, negative, single-strand, double-strand, DNA, RNA, envelope, no envelope. Okay, so this chart here, these are the viruses that you should be familiar with. So this chart here is what you should know and what you should study for some of the organisms. We will be looking at a little bit more detail at some other organisms as well, but you should know these. So you can see that it's classified here based on genome, so you can see double-stranded, DNA, and enveloped. And then you can see which families have that. So these three, herpes viridae, pox viridae. So you can see the viridae, that's what lets you know that it's a family. And then they have an example virus here of those. And you know that this is the genus because it has virus in it. Simplex virus, parapox virus, orthopox virus. And then it gives you some information as far as the clinical features. And so you should know some examples of these things. And then again, we'll be looking at some others in more detail. So you can see double-stranded DNA enveloped, double-stranded DNA naked, single-stranded DNA naked. And then we have double-stranded RNA naked, positive single-strand RNA naked, positive single-strand RNA enveloped, and then negative single-strand RNA enveloped. All right. And the more information that you'll need to know about the other ones will come as we're going through some activities and talking about the different viruses throughout this chapter and forward. So another note on classification of viral diseases. So the ICTV, remember, has the biological classification of viruses, and then it also plays an important role on classification of diseases that are caused by viruses. So in order to facilitate the tracking of these virus-related human diseases, they've created classifications that link this international classifications of diseases. So they've taken the information about the different types of viruses and they've linked it to the ICD, which is the International Classification of Diseases. And that is the standard taxonomy of disease that's maintained by the WHO, the World Health Organization. So the ICD assigns alphanumeric code of up to 6 characters to every type of viral infection, as well as other types of diseases, medical conditions, and causes of death. So this ICD code is something that is used to indicate different types of viruses or different types of diseases that viruses cause. So we have what we just went through with taxonomy for viruses. That's how we indicate the virus. And then the WHO, the World Health Organization, has gone through and they've given some codes to the diseases that these viruses cause. And this paragraph here is just saying that this number is important because then it's used in the medical profession. Medical coders and insurance people use these different codes to know what kind of organism they're working with, to know what kind of tests in order to run, and what kind of tests that they will cover through insurance, for example.